This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 417, recorded on November 25th, 2016. This episode is brought to you by CuriosityStream, a subscription streaming service that offers over 1,500 documentaries and nonfiction series from the world's best filmmakers. Get unlimited access starting at just $2.99 a month. And for our audience, the first 2 months are completely free if you sign up at curiositystream.com/microbe and use the promo code microbe. Hi, everybody. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Fort Lee, New Jersey, Dickson Despommier.
Hello, Vincent. Happy Thanksgiving Day after.
You too. Where the hell is Fort Lee?
Come on, you know where that is. It's where Mr. Richard Fader lives.
Isn't that where Bridgegate took place?
It certainly is, and I was part of that, too. I was stuck in traffic all 3 days.
That's right. It was a thing to get back at the mayor of Fort Lee, right?
You bet. See how well that worked out.
Yep. Also joining us from southeastern Michigan, Kathy Spindler.
Hi, everybody.
Happy Turkey.
Hello, Kathy.
Thanks. So today it's 42 degrees Fahrenheit, 6 degrees Celsius, gray. Not too much else to say about it.
Dickson, were you here in my office, you would look out the window and almost not be able to see New Jersey. It's completely cloudy. Of course, it's getting dark. It's 3:13. It's getting dark. It's 11 Celsius, and it's raining. Is it raining over there?
Not in Fort Lee, not right now.
And you're maybe 3 miles from me or less, right?
I'm a little bit more than that, I think.
How wide is the Hudson here?
How wide is the Hudson? That's a good question. It's almost a mile.
All right, so you're not 2 miles. You're 2 miles from me, not more than that, because I can see your apartment from here. In fact,
where are you calling from?
I'm in the office in Hammer.
Oh, I thought you were home.
No, I come here to get good sound.
I got it.
Also joining us today from Austin, Texas, Rich Condit.
Hi, everybody. How you doing?
Hey, Rich.
Do they do Thanksgiving in Texas?
They do, except that as part of the traditional Thanksgiving meal, we have tamales.
But right. you also have turkey, right?
We also have turkey, and in this household being my daughter and son-in-law, we have ham too.
Do you deep fry the turkey?
I was going to ask.
Do you barbecue it?
Don't do that. No. Traditional roast the turkey thing.
All right. Did the rest of your family come, or is it just that family?
Just this family.
That must be.
We've gotten, with the kids, everybody's got families and that kind of stuff. Traveling over Thanksgiving is just such a rat race.
It is.
That we just kind of given up on it, right?
Yeah.
Travel other.
I returned from Montana on Saturday, and every seat on every plane was full. It was so crowded everywhere. Yeah.
Supposedly, this is the most traveled Thanksgiving ever in history for the United States.
Why is that?
Don't know.
Because the world is about to end. It may be the last.
They're all going to each other's homes to say goodbye.
Yeah.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here at last.
Hey,
for bringing me in.
Sorry.
It's all right. just sitting here holding my tongue. It's foggy and gray here and overcast, low overcast, and getting dark already. But,
I'm only slightly better than you guys. We got 65 degrees Fahrenheit, 18 C, and cloudy. Okay. But at least I got sunshine. But of course, I'm in Central Time here. So,
right.
Kathy, are you in Central Time?
Nope.
Eastern.
Detroit is in the Eastern time zone.
Yep.
You've never heard that in the Detroit
That's
airport?
That's right.
I say it all the time because people think it's going to be Central Time. Yeah.
Eastern Time. The Department of Microbiology at the Icahn School of Medicine at Mount Sinai wants you to know about their graduate and postdoctoral programs composed of over 20 virology labs, all centralized in one building in the heart of New York City. This department's a perfect fit for anyone with an interest in pursuing virus research. The department is presently looking to recruit any prospective graduate students to apply to their program by the December 1st deadline. Interested postdocs are also encouraged to contact faculty of interest. For more information about the department, check out bit.ly/MICROMSSM. It's all one word, bit.ly/MICROMSSM. To everyone who celebrates Thanksgiving, happy Thanksgiving. It's the day after. It is, what is this, Black Friday?
Black Friday, yes.
Black Friday.
And Monday is Cyber Monday.
Right.
And next Thursday is World AIDS Day. Did everyone know that?
Amazing.
We do now.
It was one.
It was a day for everything.
It was the first Infectious Disease World Day.
Oh,
yeah. If you go to worldaidsday.org, it will tell you that. And in our paper this week is in honor of that, I suppose, but not intentionally. It's an HIV paper. But before we do that, a very brief follow-up. The WHO last week made a statement that Zika virus and its associated consequences remain a significant, enduring public health challenge requiring intense action, but no longer represent a public health emergency of international concern. And there is a statement which we'll put a link to in the show notes that explains all of this.
So it's no longer an emergency. It's just another crummy thing that we have to deal with.
Yep, forever and ever.
Forever and ever. It's gone into the chronic phase now.
It reminds me of what happened when West Nile virus first came to this country. Everybody was not freaked out and panicking, but they were certainly on high alert. And the next year it was a little bit less, and the next year it was even less than that. And today it's just like, what happened to West Nile? Did it go away? And the answer is, of course not. It's all over the place.
Well, we get kind of an annual report. I know I hear about the West Nile virus cases that happen, and it's just one of those things in the background.
That's right.
Mm-hmm.
So, at the risk of opening a can of worms, in the popular press,
we never stop.
In the popular press a couple of weeks ago, there was a couple of articles about how the apparent incidence of microcephaly in Colombia was less than in Brazil, and this sort of rekindles the idea that there may be some sort of cofactor in Brazil in the microcephaly. What has, and I looked at the numbers that they had, and, on the surface, the numbers seem to support that. But of course, it's got all, what is it? There's lies, damn lies, and statistics. It's got all of those problems. Do any of you know anything about this?
I do not. I don't think any sizable study has yet come out that would address it. And that's what you really need, not anecdotal numbers. So I would wait. We'll know, right?
Well, and we need numbers that we can actually compare, because there have been lots of record keeping problems, and I know we commented early on in the Zika epidemic that the microcephaly epidemic that was supposedly happening in Brazil was really kind of in the ballpark of the rates of microcephaly that you see ordinarily in other countries, and maybe it was just people were noticing it. And we now know that there are mechanisms that would link Zika to microcephaly, but whether that's a huge common mechanism, it's still not clear at all. And the Colombia thing, it's going to have the same limitations, I suspect.
Sure. The other thing I heard about Zika recently was that a normal, of quotes unquote, normal birth, the head size was normal, that following that, after birth, the patient can develop microcephaly. And I thought that was very strange to hear. Does anybody hear anything about that?
Well, I've heard that there can be neurological issues that are not obvious at birth as microcephaly. We've been suspicious that might happen for a while, and I guess there's some evidence for that now. That doesn't surprise me a bit.
Right.
I think there was a story either in yesterday or the day before's New York Times about some twins in Brazil, and I think one of them maybe at birth had microcephaly, and then some months later had microcephaly. Or anyway, there was mention of twins, but also of microcephaly not defined at birth, but appeared later.
Maybe that's what I was referring to.
All right. That wasn't much of a can of worms, Rich.
Yeah, that worked out okay.
You'd have to go on TWiV for a real can of worms there, Rich.
Yes.
Well, maybe you need to go to what is it? The livingriver.org?
That's right. I know you only fly fish. We don't use worms on.
Speaking of fly fishing, let me relate a little story from my trip in Montana.
Oh, yeah.
I went last week to Hamilton, Montana to visit Rocky Mountain Labs. And Thursday evening, I attended a meeting of, what is it called, Dickson?
Trout Unlimited.
Trout Unlimited.
Ah.
It was mainly old men listening to a talk by a young woman, actually, who works in Missoula. She works at the university and also with the Department of the Interior. And her talk was about eDNA, environmental DNA. And what they do is they collect water from streams and they do PCR for specific fish. And what they do is they look for mitochondrial genes and they can tell what fish are in the stream.
That is amazing.
Wow.
It's really amazing.
And she gave a talk to this audience about this. And it was very interesting because the way they previously determined populations of fish, and she was mostly talking about bull trout, which are endangered there, right, Dickson?
Mm-hmm. Yep.
They shock the water, and then the fish get temporarily stunned. They float to the top, and they count them.
Mm-hmm.
Right.
And she showed some data which shows that this is much more sensitive than that. You can detect way more fish, and you can get populate. It's very interesting. But, they filter 5 liters of water from a particular location. They filter it right there on site, and then they put the filter in a bag and take it back to the lab to do some extraction and PCR. So in theory, they could look for a lot of different things.
That's true.
Since they don't do deep sequencing, but that would be interesting. They just do PCR to target a specific species.
Right.
But
the one thing you're going to not get from that is the size of the fish, which you would get when you electroshock.
The size of the fish and the number of each one.
And
right?
males and females and that sort of thing. So although they could, I'm sure they could, if they wanted to, they could probably extend that study.
Well, if they, there might be a way to make it quantitative. I don't know. Especially if
you did deep sequencing, you might be quantitative because of the number of reads or something like that.
Yeah.
So the thing that was interesting, she said when fish are spawning, you get a lot of, she kept saying this, you get a lot of high quality DNA.
I imagine so.
She said that over and over again.
Well, because
is that because that DNA needs to be stable? I mean, because
they don't have internal fertilization; it all occurs outside.
Right. That's what
I think it's recently released. That's why. Whereas when the other DNA they're looking at is coming off the skin.
Right.
Exactly.
Right. So it may be old.
Exactly.
Right. So the DNA they're getting during spawning is in cells that have evolved to survive in the water. Getting to Kathy's point.
Bingo. Bingo.
High quality DNA.
High quality DNA.
It was fun. It was at the local Elks Club,
Good stuff. Serve beer.
They had a bar. They were serving beer. Yeah. I must say, Dickson, I had a little trouble staying awake. I mean,
that's what the beer is all about.
She actually was excellent. She presented it in a way that was perfect for the audience, but I was getting bored.
Yeah.
She didn't mention PCR once, and at one point someone asked her a question. She says, "Okay, I'll have to use this term, real-time quantitative PCR, if that means anything." Then I woke up.
Oh yeah.
But it didn't have anything to do with the time change that you were sleepy, right?
Oh yes, absolutely. Because this thing started at I think 7 p.m., so it was already 9, and it was an hour and a half.
Wow.
And I just wanted to get back to my room. I had some things to do for the next day, and it wasn't all that interesting. She dragged it out a bit. Could have been an hour, but it was cool that you could use this. I just kept thinking of Eric Delwart. DNA is everywhere. It's a real problem.
DNA is everywhere. It's really a problem.
It covers the earth, so to speak.
But Rich, before you and I went and talked to him, I hadn't thought of this, that DNA is everywhere, right?
Yeah.
But obviously it is.
Yeah.
Things are dying left and right.
That day is so fresh in my mind. That was a wonderful day.
Yeah, we had a good time. We're talking about an old TWiV where we visited the Blood Systems Research Center in San Francisco, and our host was Eric Delwart, and Rich and I went. It was after a study section, I believe.
Yeah, and we went out to lunch with him, and we walked around the city some, and he would look down alleys and tell you it was a great source for DNA.
Yes. It's a different perspective on life. Yes.
Dude.
All right, let's do a snippet. We have a real snippet today. This is a letter published recently in Nature called 1970s and Patient Zero, HIV Genomes, and the Patient Zeros in quotes, illuminate early HIV/AIDS history in North America. And this is from Worrelby, Watts, McKay, Souchard, Grenade, Toywin, Coblin, Heinen, LeMay, and Jaffe. They're from University of Arizona, University of Cambridge in the UK, UCLA, and CDC, someplace in Belgium called the UCB, Laboratory of Infectious Diseases here at the New York Blood Center, and the Department of Microbiology and Immunology at the Rega Institute in Belgium. Now, as I said, this Thursday, December 1st, is World AIDS Day. And AIDS is a disease, of course, caused by HIV-1 that became recognized initially in 1981, virus identified a few years later, and we now understand from many people's work, including Beatrice Hahn and others, that the virus crossed over from a monkey probably in the early 1900s as SIV, infected a human, and then spread throughout Africa and then went elsewhere. The earliest samples we have from which sequences were derived, I think, were 1959. These are in 1960. These are two 1959-1960 samples, which allow us to date the spillover to the early 1900s. But considering that, the epidemic first emerged in the U.S. and spread extensively in the 80s and beyond, we don't have any sequence from early U.S. isolates. So that's what this paper is attempting to get around. Can we have... And now, we have lots of archived serum samples from 1978 to 1979. They were originally collected from men who have sex with men. These are cohorts in both New York City and San Francisco. So these are stored serum samples that are really a valuable resource. So in this paper, they tried to... first, they did a really interesting experiment. These are, let's see, New York City sample... 378 of whom had been an earlier cohort of 8,900 men involved in hepatitis B virus screening. Previous work had showed that 6.6% of these were seropositive, and they picked some of those for sequencing. And then from the San Francisco cohort, 6,875 patients also enrolled in HBV studies, which is clearly a sexually transmitted infection. They tested 2,200 of these by Western blot to see which ones were positive for antibodies to HIV. Can you imagine 2,200 samples tested by West? That's a lot of gels, right?
Yeah.
Wow.
True.
And then they picked 20 of those for sequencing. Now, these are old samples, and the RNA is degraded. They say the RNA is only in short fragments. So they wanted to amplify this DNA by PCR, so they developed what they call an RNA jackhammering approach, which they basically use a lot of primers to amplify very, very short fragments of RNA because the RNA is fragmented, right? So you're doing a lot of PCR, and then you have to overlap the sequences, which is done computationally. But they were able to get 3 samples from San Francisco and 5 from New York City, complete coding sequences from the genomes. And so this is all new, early 70s, 78, 78, 79, late 70s, but earlier than anything before. And so this is group M. HIV exists in 4 groups, M, N, O, and P, 4 different spillovers from monkeys into people. M is the big one that causes most of the infections in the world. So these are group M sequences. And they can tell a number of really cool things from this. First of all, that this virus in New York arrived via Haiti. So it came to Haiti in the let's see, in about early 70s. Came
69
from
to 74.
69 to 70 is the range.
If I read this correctly.
Yeah. And then before Haiti, it came from Africa. So it went from Africa to Haiti to the U.S., and people have wondered whether it went from the U.S. to Haiti. This sequence analysis, and by the way, I'm not talking about exactly what they did, but they do a number of analyses, including phylogenetic tree construction and others. So Africa, Haiti, 63 to 71, New York, 69 to 74, and then from New York to San Francisco. And those are the 2 epicenters from which the virus diversified, and then it spread elsewhere in the U.S. and elsewhere globally as well. And so the New York epicenter was first, and then San Francisco epicenter was established later. So that's pretty interesting that we can get that information, which we really didn't know. Another point, the New York City epidemic was already diverse by 1979. So it had been circulating for a while, as we said, 60 to 70. That's really interesting. That's the main finding. Now, the other one is kind of incidental. Everyone probably has heard of a patient called Patient Zero, and that person was identified in Randy Schilt's book, and the band played on. In fact, they mentioned the book in this paper. It's not usual that you have a book mentioned in Nature.
Right.
Mm-hmm.
But he identified Patient Zero as a highly sexually active French-Canadian flight attendant, Gaetan Dugas. If you just go to Wikipedia, you'll find his name there, so it's not like we're revealing anything. He's long dead, of course. But in California, CDC investigators were trying to put together a transmission graph of 40 men, a part of a sexual network, and they were trying to figure out who was spreading virus to whom. And one
a typical sort of epidemiologic thing where you try and figure out the source of an infection.
Right. Tracing the contact tracing, right? And this was a known sexual network. These people were interacting with each other, so it's not like they had to do a lot of digging. But they put one person at the center who seemed to be at the center, and everyone was had contact with him and got infected via him initially. And they, according to this paper, they called it Patient O because he was outside of California, right? And others were inside California. And then later, this got erroneously transmogrified to Zero. So they called it Patient Zero. And then, in epidemiology, Patient Zero is the original or the primary case. And so in Schilt's book, they say he was the first case in the U.S., and he spread virus to many other people because he was so sexually active and he was traveling all over the world and so forth. So if you go to I'm not sure this is even right. If you go to Wikipedia, you can download this chart, and it doesn't have O in it. It has Zero in it. Now, maybe it's not the original chart from the paper, so I'm not sure how accurate this is. Anyway, they have some samples because he was very good about giving samples. There's lots of serum stored and so forth. So they sequenced his genome as well, and he is not, on a phylogenetic basis, he's not before everyone else, essentially. He's just part of the early diversification of HIV in the U.S. So what they say, he was not basal to the U.S. diversity, let alone to the deeper Caribbean subtype diversity in a manner that might be suggested of a special role. So we find no evidence that Patient Zero was the first person infected by this lineage of HIV.
Right. He was obviously linked to the group that he'd been linked to by the CDC, which was those few dozen people. But that's not, that's certainly not Patient Zero for an epidemic. And really, though, there was evidence to that effect before this paper, but what this paper adds is the phylogenetic analysis that traces this back and gives us really definitive signposts. I think the Patient Zero story is the thing that got picked up in the press for the most part.
Yeah, right.
Understandably, because it's a human story and it's something that a lot of people never got the memo that wasn't really Patient Zero.
And is this the first time that we have the information that it's because of O and Zero getting confused?
I think,
for me,
the first I've heard.
Yeah. That was news to me. Yes.
Yeah.
But I don't think that that's news to everybody. Okay, because I think it sounds to me from reading this that there was suspicion that this whole notion that he was Patient Zero, and it even says somewhere here that people have corrected people who gave the original nomenclature of patient O have corrected people on numerous occasions, but the story is so attractive.
Nobody listens,
right? Yeah. Sexually active flight attendant from out of the country, okay, who comes in and spreads this all over the place. I mean, how can you not get hooked on that?
It's just dripping with truthiness.
Yes.
Yeah, right. So I have I went to some of these links to find that what they say is this original figure from the paper. So there's a Wikipedia story that's linked through the virology blog I wrote on this. And they have this case tracing, and they have 0 in the middle. It's clearly 0, not O. And it says source, American Journal of Medicine, March 1984. So I don't know if this is the original figure or, a version that was drawn where they made the mistake already. Anyway, it's pretty interesting.
Is this the same? Are you talking about the same chart as they show in this? It's your virology blog, of course. Okay.
Yeah, so I took that from a Wikipedia page on the origin of AIDS, and actually Dugas, I think, was the page I searched for, Gaetan Dugas, and I don't know if that's the original figure from the paper or something that someone drew afterwards and made the mistake. Okay, so I didn't look at the original paper, but someone will.
The up at the top of the chart, it says the source is the American Journal of Medicine, March 1984.
Right. And that's reference 5 in the paper. Here we go. I got the paper up. Oh, so they redrew that because it's in black and white. I can't tell if it's O or 0. Sometimes it's hard.
Yeah.
I'm going to put the link to this in the show notes and you could look.
I've downloaded the PDF here.
You got it. You can see it's the first figure. And
it's... Well, it's hard to believe that it's not 0 because every other thing is a number.
I know, right. So where does this O come from? That's what I'm right. All right, that's all. Anything else? I think it's a cool.
So we don't know the story of O.
We don't know the story, but clearly Dugas was not. Dugas was not. Oh, that's a great title. The story of O. We'll get a lot of hits.
So actually, at the top of this figure, they have. 0 equals index patients, 1 through 21, sequence of onset.
Right.
Keep talking.
It looks like a 0, doesn't it?
Keep looking through.
They're all numbers. I don't know why.
I think it's 0. Yeah.
But
right, 0.
Despite this uncertainty, what's clear is that the virus went from Africa to Haiti to New York to San Francisco and then elsewhere, and Dugas was not patient 0. He's not the first person in the U.S. to be infected. Right. There might have been someone, but we don't know who it is. Right. Anyway, it's pretty neat stuff. I think technologically it's amazing that they could get full-length coding sequences out of this, and it's all fragmented. So
yes, the real story of this paper, scientifically, is the technique and the tour de force of the sequencing that they did.
So now it says if you have old samples from at least the 70s, you could get some RNA virus sequence out of it. So don't give up. What is it? RNA jackhammer. This episode is brought to you by CuriosityStream, the world's first ad-free nonfiction streaming service with over 1,500 titles and 600 hours of content. It was founded by John Hendricks, formerly of Discovery Channel. What does that mean? You're guaranteed access to real science shows. Now, CuriosityStream is available on many platforms. You could just open a web browser and go to their site and watch the content, or you could watch it on your TV using any of those devices that interface the internet with your TV, like Apple TV, Roku, Chromecast, etc. It's available in 196 countries worldwide, and what you get is a wide selection of science, technology, nature, history, documentaries, interviews, lectures, a lot of stuff, and I think listeners of TWiV and other shows that we do would be interested in their content. For example, they have Stephen Hawking's Favorite Places. It's a brand new documentary where Stephen Hawking pilots a fantastical CGI spaceship across the universe, stopping at his favorite destinations. Digits is a 3-part series hosted by Derek Mueller, creator of the science channel over on YouTube called Veritasium. And he explores online safety and security, has interviews with former NSA Director Edward Snowden and co-founder of the internet, Vint Cerf. Deep Time History, a 3-parter on the 14 billion year history of the universe, underwater wonders of the national parks, what's going on under the bodies of water there, and much, much more. Plus, they also have ultra-high definition content, so-called 4K libraries that have over 50 hours. You can get monthly and annual plans to watch CuriosityStream, And they start at just $2.99 a month, less than a cup of coffee. Check out curiositystream.com/microbe and use the promo code microbe during sign-up to get unlimited access to the world's top documentaries and nonfiction series, completely free for the first 60 days. That's 2 entire months free of one of the largest nonfiction 4K libraries around. Just go to curiositystream.com/microbe and use the offer code microbe at sign-up. We thank CuriosityStream for their support of TWiV. All right, let's do some email.
Right.
Jason writes, greetings, VRADK. I leave it to you to figure out the greeting. Not so hard.
Not so hard,
actually,
exactly right.
It sounds like a peptide of some sort.
Yeah, it's highly immunogenic.
Exactly.
Vincent, Rich, Alan, Dickson, Kathy.
There you go.
Is that all of us?
Even I got that.
VRADK.
There's no anonymous chupacabra.
There's no anonymous chupacabra.
No.
Chupacabra. All right. I'm a bit behind
Which,
on TWiV.
for the audience which didn't hear the pre-show, we're in Google Docs. Apparently, anonymous under certain permission settings, somebody shows up as an anonymous animal, and Dickson during this show is anonymous chupacabra. He was originally anonymous hedgehog. So
that's right.
Now you're all in on the joke.
I'm going to call you that. Hey, good morning, Chupacabra.
You better watch out, or I'll get your coat.
I'm going back to the letter from Jason. I'm a bit behind on TWiV, but I just listened to episode 411 and heard about the final book contest. I realize you've likely picked a winner now, but I couldn't help but submit this for your enjoyment. I have a feeling Alan will enjoy it. There once was a virus named Vincent who appeared to all as innocent, but as it turned out, as it floated about, it mutated into something magnificent. I iterated a few times on this and waffled between using Maleficent and Magnificent.
We all feel that way about Vincent.
Yeah. The former sounded interesting, but the latter seems more appropriate for TWiV. Maleficent is okay. I've been listening since the first Drobo contest, and despite not being in the scientific field, I find your podcast quite informative. TWiV holds a place within the list of other podcasts I listen to on the 3-hour commute each way I take twice a week to head into the office. I'm so sorry. I thought it might be interesting to list some of the other podcasts I listen to as well. He listens to TWiV, Nerdist, Penn's Sunday School, starring Penn Jillette of Penn and Teller fame, Smodcast, starring Kevin Smith and Scott Mosier of Clerks fame. I don't know what any of this means except Penn and Teller.
Oh, you don't know? You haven't seen Clerks?
No, don't know what it is.
Oh,
what is it?
It's a movie.
It's a movie?
Kevin Smith is a director. Clerks was his first movie. You should definitely see Clerks.
Defensive security, on the media, note to self,
right? And
when there are new episodes, We're Alive, which is a radio play. Definitely varied, but it keeps the queue full, and I'm never wanting for something to listen to. Keep up the great podcast. It's wonderful to see you expand your viral empire and bring new podcasts into the fold. Science communication is essential, and I find the information I learn on TWiV to be useful in many ways. Thanks. So I thought it would be interesting to hear from our listeners what's on your podcast list.
Yeah.
Does anyone on TWiV listen to other podcasts?
I do.
What do you listen to?
I listen to Away with Words.
Oh, that sounds good.
Yeah, and then, of course, some of the other in the Twix family. I have Masterpiece Studio, and then things if I miss them on NPR, like Wait, Don't Tell Me or Radio Lab. Trundlebed Tales I used to listen to, but I haven't in a long time. I have Public Health United, but they don't come up very often with new ones. So, yeah, those are them.
Yeah. Now, I'm not going to ask Dickson because I know Dickson doesn't listen.
Not as much as I should. That's true.
Any podcast.
Yeah, that's right.
Do you listen to any, Rich?
Not really. I have, off and on. I did, what was that? Hardcore History.
Yeah.
For a while,
right.
And I did the series on the murder case. I forget what that was called. That was really hot for a while.
Serial.
Yeah, Serial. But not so much anymore. It'll be good.
Alan, do you listen?
I'm kind of like Rich. I'm sporadic about it. I'll get into a podcast and listen to episodes for a while and then not. I listen to the TWiVs that I'm not on so that I know what the joke is next time.
Yeah.
But I don't have a commute usually, and I don't really have a spot in my day when I would do that, so it doesn't really fit in at the moment.
I must say that my earbud time has been sucked up seriously recently with Kathy's book series that she sent me. I'm on book 7, Kathy.
Okay, so am I.
I can't stop.
I know. Yeah.
Well,
I listened to her in France twice. Book 6.
Yeah.
Listeners, send in your list of podcasts. What do you listen to? Let us know. Rich, you take the next one.
Oh, I got to go back there. I've been looking through this paper. These guys are from the CDC who published that paper.
That's right.
And it's clearly they're calling him patient 0, but they talk about previous CDC surveys. So if as far as I can see at this point, if there's any guilt to be found in transmogrifying this to patient 0, it's these guys. All right. If in fact it was patient O before.
Previously, yeah. Okay.
Sorry. Okay.
That's good. Yeah, it may have happened before. That's it.
All right. I'm on Saranga. Dear TWiV professors. Actually, that's interesting. I have a friend from Sri Lanka named Sarangi. So I wonder if that's it. Well, at any rate. Greetings from Sri Lanka. I'm a medical virologist from this beautiful island nation with an eternal summer who has been following your vibrant podcast for the last 5 years. I'm writing you this email to express my gratitude for your service rendered in science education, especially in virology. Your podcasts have been my inspiration during my commute to work through an ever-growing and ever-slowing sluggish traffic in Colombo, which makes the waiting in traffic an enjoyable one. Virology, either as an applied or basic subject, is not well developed in this part of the world, where we have only a handful of people with some expertise. Certainly there are exceptional and brilliant people such as Professor J.S.M. Piras, FRS, who is a Sri Lankan with immense contribution to the field of medical virology. But generally, the rest of us need to work to have a constant motivation to improve our knowledge of virology. I find TWiV as one of those motivational factors in my virologist career, and I believe a few of my colleagues do feel the same after introducing them to your podcast. Your podcasts orient the listener in the direction of new developments, as well as in the breadth of the field of virology as a scientific, artistic, and a cultural subject. Let me tell you a bit about our work in virology in Sri Lanka. We are a group of medics who are involved in the diagnosis and management of infections in Sri Lanka. The field of medical microbiology has been in the country for the last few decades, but our subgroup has emerged recently within the last decade. Sri Lanka is, in fact, one of the few countries that has the separate field of medical virology as a medical postgraduate subject, among other countries such as UK. Currently, our tasks span from laboratory work, the job of clinical scientist and a pathologist, to the clinical work, the job of an ID physician. We are a middle-income country with a totally government-funded universal health care system.
Rub it in.
Yeah, we do have a small and growing private sector. The field of virology is at a lower level of development when compared with the developed world. We are gradually trying to expand the services, which are hampered by the limitations of resources, but we are slowly improving. The landscape of viral infections in this country is vastly unknown, especially with reference to arboviruses and other zoonoses. For most of the viral illnesses, there are limitations in diagnosis as a nation, except for the common ones such as bloodborne viruses, for example, HIV, common arboviruses, example, dengue, and common zoonoses, for example, rabies. On the other hand, in clinical practice, we do have limitations in available therapeutics for viral illnesses, and sometimes we have to adapt to survive with limited resources. A good example is the rabies vaccination in post-exposure prophylaxis, where we follow vaccination via the intradermal route instead of the intramuscular route as a cost-cutting but effective measure. I believe practicing clinical virology or even scientific research, basic or applied, is a different form of art in the developing world. Among your diverse podcasts of TWiV, you have discussions with many groups of personnel, such as medical personnel, international groups, etc. I would like to propose you do some podcasts about virology, microbiology, and the resource-poor settings, too, if it is feasible. Wonderful idea.
Yes.
It is my privilege and pleasure writing to you. Looking forward to listen to many more Twix podcasts. Live long and prosper. Saranga, who is an MD. P.S. Current weather in Sri Lanka has turned into a moist and wet one from a sunny and dry one due to the recent activation of the northeast monsoon, and the temperature now in Colombo is 31 C.
Right.
A cloudy sky. Whoa.
Isn't Colombo the city in which Arthur C. Clarke lived as well?
Maybe.
I believe so. He was in Sri Lanka.
Yep.
Yeah, I think he was in Colombo. Right.
Have you been, Dickson?
No.
Has anyone been to Sri Lanka?
No, but I've been looking at pictures of it all the time while Rich was reading.
It has so many endemic species that, the chameleons, for instance, are world famous for the Sri Lankan evolutionary biology. Really interesting stuff.
I think this is a good idea, talking with virologists, microbiologists in resource-poor settings. We never do that. Of course, we talk about them in Europe and South America, Brazil, U.S., but,
Well, it looks like we have
we should do
it. I always tell people that if you're going to make a suggestion, be prepared to volunteer to carry it out. So maybe Saranga is a volunteer.
Yeah. Well, I don't know how Skype would work, but that might be challenging.
Well, it would. Other than the time zone, I've interviewed people in many parts of the world, and if they have a working internet connection,
it's okay.
It's usually doable. That sometimes you get the delay is longer. You may have more cutouts depending on how the signal's being routed, but it should be doable. The time difference would be
that's the problem. Yeah.
It would be interesting.
So Rongo would be getting up at 3 o'clock in the morning.
if we could establish a network of sentinel virologists throughout the world that report regularly to TWiV, and this is a great start. This would be terrific.
Mm-hmm.
Yeah, maybe a good idea. All right, Alan, please take the next one.
Sure. Gopal writes, "Hello, Vincent. I seem to recall you have occasionally mentioned on TWiV in a mildly disapproving tone that your kids might pick up some microbes from their dog. So when I came across this paper in an Elsevier media alert, I thought I should bring it to your notice. The paper is Companion Animals as a Source of Viruses for Human Beings and Food Production Animals from Journal of Comparative Pathology," and gives the link. And Gopal says, "The paper, I'm glad to say, is open access." So there you go, Vincent. You're absolutely right.
It's a nice, interesting paper. They looked at a variety of pets and food production. Actually, this is a literature review.
It's a literature review.
Scanned the literature, and they found 138 viral species capable of infecting companion animals. Now, they do have these nice pie charts here where you can look at viruses from food animals or from your pets. So let's see. Viruses of domestic carnivores. That would be dogs mainly, right?
Dogs and cats.
Dogs and cats, that's right.
So viruses, zoonotic viruses, 59. Then they go horses, dogs, mammals, birds. Interesting. Yeah, there are a lot of them out there. I have to say, the other day, my dog sneezed in my face, and I wondered if I would get sick, and my wife said, "No, I got her the flu vaccine." So, okay. What about all that other stuff lurking in her
pharynx? Have you had your distemper shot, Vincent?
Yeah, right. I'm okay. It's been 2 days.
Good.
You could have come down with cholera.
Oh, really?
That was
cholera.
Cholera.
Oh, cholera. It's not a collie.
Cholera. Well, I don't know what kind of dog you have, but you know.
I know. You don't listen to me when I talk.
This is all true.
This is a cool paper. Anyway, there are lots of...
Well, at least you didn't get the Shih Tzus.
Shih Tzus. No, we didn't get a Shih Tzu. That's absolutely right. We don't have a Shih Tzu. They bark way too much. Anyway, there are lots of viruses that infect not only food production animals, but your pets. So check it out. Thank you, Gopal. And Alan, you want to take another one?
The Secret Viral Life of Pets.
Do you want to take
Yeah, I'll go ahead and take the next one from our friend Wink, who writes, maybe this is the first highly successful gene therapy. It's for X-linked severe combined immunodeficiency syndrome, SCID, commonly known as bubble boy disease. Lymphocytes from children with SCID are transfected ex vivo with a gamma retrovirus containing the human adenosine deaminase gene, which is what's lacking in SCID. The therapy was approved in Europe under the name Strimvelis. While SCID is uniformly fatal, usually in the first year of life, 15 of 18 subjects were apparently cured.
Wow.
Yeah.
This
yes, there's the link.
This is an old study, 10 years ago, and it was notorious because, so what they do in this, they take bone marrow stem cells, they infect them, and these kids have a deficiency that leads to SCID, and they replace the gene in the bone marrow cells in culture with a retrovirus, and they put the cells back in the kids, and they repopulate them, and it takes care of their SCID. The problem is initially a number of these kids within 2 years, I think, developed leukemia because the retrovirus integrated next to an oncogene.
Right.
And that, I think, caused cessation of this kind of gene therapy for a while.
Right. They said they cured 3 out of 4 of those.
Yeah, those kids ended up being okay. And then they changed the vectors subsequently so they wouldn't do that. But it's quite, this is a 10-year follow-up to that.
Right.
It's quite an interesting trial.
It's pretty impressive.
Yep.
So is this therapy still going on with different vectors? Because I mean, really, even with the problems, 15 out of 18 subjects apparently cured, or at least out of 10 years is pretty good.
Strimvelis, which is now from GlaxoSmithKline.
It's licensed?
Got European licensing in May of 2016, May of this year. And
what's that? Is that a
that's the commercial version of this therapy. It's an ex vivo stem cell gene therapy. It's the same strategy.
And have they addressed the integration problem?
I'm just looking through now.
I don't see how you can, really.
I think they. I asked Steve Goff this a few weeks ago, and he said the vectors have been modified to make them less integrative. But you do want it to integrate, obviously.
Yeah.
But not. Yeah. I don't know how you would address it, but this is not an issue anymore.
So they had a 100% survival rate at 3 years post-treatment with Strimvelis in the pivotal study that led to the approval. That was 12 patients. And every child receiving the treatment who contributed to the marketing authorization data package is alive today. That's 18 kids.
That's amazing.
It is.
That's amazing.
Could you, the way this is done, you culture the bone marrow stem cells, you infect them, right? Then you put them back in the patient. Could you, before you put them back in, sequence them and see where the integration sites are? Or is that like way too expensive to do that?
My guess is that this is not a clone.
No, it's not a clone. They're multiple. Right. They're different insertion sites, right? So you could clone it and then say, okay, this insertion site is not next to an oncogene, but I don't think they do that.
Clone. Right.
I don't think they do that. I think they just have modified the vector to get around this, and I cannot remember what was done to it. I'm very sorry.
This is this was one of the first targets for gene therapy because it's kind of, as diseases go, low-hanging fruit, because the appropriate therapy is ex vivo, and bone marrow transplants, people know how to do that.
Yep.
And it can be cured simply by supplying a missing enzyme. In this case, adenosine deaminase is involved in nucleotide metabolism, and it messes up DNA replicate. The lack of it messes up DNA replication in these cells, which causes the immune deficiency. And all you have to do is supply that enzyme.
And the final critical factor that made it an appropriate target is that if you don't treat it, or if you use current treatments, this is a uniformly fatal disease.
Right.
Yeah.
So it's not likely that you're going to do any worse than what we already had.
That's right.
So the nice thing here is that you don't have to infect people with a virus because it would be hard to target the right cells. You take them out, you infect them in culture, then you put them back in. Because they're stem cells, they will start to give rise to other cells, and they're always there.
Yeah.
So that's a nice... You can't do that for everything, but when you can, it's a nice approach.
Definitely.
I hadn't thought about this for a long time. It's just really remarkable. And I didn't know about the updates. This is great. It's remarkable. That's amazing.
Yep.
It is.
Look at what we can do. That's
Look
amazing.
what we can do. All right. Well, Dickson, you are next. Phil.
Okay, then. Phil writes, "Hello, Vincent. Are bacteriophages not classified by the Baltimore system?" Thank you, Alan. "According to the wiki, there are 19 types of phage which they list. I follow all of your podcasts and started to do your online virology course. Thanks so much to you and your teams for all your hard work and dedication. I live in Milton Keynes, England. Thanks again." Then there's a follow-up.
No, that's me, Dickson. Don't read.
Oh, okay.
That's me. I forgot to add my name.
There's no follow-up.
But, I mean, they are, of course, the genome is... Dickson, how many different genomes are there?
Seven.
So they can be placed in the Baltimore system for sure.
Yes, they can.
But if you go to Viral Zone, which is a lovely resource...
That's the first thing I did of reading this.
So, for example, you click on double-stranded DNA viruses. Amongst all the other viruses, the phages are in there too.
There you go.
You got your herpes viruses, which are, of course, human herpes virus and animal herpes virus. But then right below it, the caudovirales, which are a bunch of different phages, and they're all mixed in. So, bacteriophages are accorded the status that they deserve in the viral world. All the world's a phage, actually.
Yeah.
So the Baltimore system, when David first proposed that, was that as a publication? Did he say we ought to do it this way?
Yeah, he published
and I wonder
paper,
if in that publication he made any distinction between animal and bacterial viruses. Because this is a good question, because I've always kind of blithely thought of it as in terms of animal virology, but clearly it can, and in Viral Zone does incorporate phages.
Well, let's see. It was Bacteriological Reviews in 1971.
Whoa!
The name of the article is "Expression of Animal Virus Genomes," and you can. Of course, I'm getting it, but it doesn't mean that you will be able to. It's David Baltimore. "The world of animal viruses appears to offer an unfathomable diversity of specimens. But as the molecular biology of the replication has been studied, a pattern of behavior has emerged." And he said you can divide them into classes, each of which has its own method of course, this is not written properly. A class doesn't have its own method of transmitting its genetics.
Do viruses have behaviors?
No. It's all messed up, but that's okay. It's a good...
I'm fine with viruses having behaviors, but I know some people aren't.
So let's see. He had, of course, 1 through 6, because he didn't have the hepatinoviruses. But let's see if any phages influenced him.
Well, in the title, he's talking about animal virus.
Animal virus.
Yes, it's animal virus genomes, yeah.
So is it definitely the bacteriological reviews that's the first one? Because I just put in Baltimore D virus 1971, and there's other things that don't have animal virus in the title, like viral genetic systems.
With Baltimore in it?
Yep. Yeah. Trans New York something or the Academy of Sciences.
Now, this is the one that we referenced.
Okay, viral genetic systems.
Yeah, these are certainly animal viruses, you're right. But, of course, the phages can fit neatly into this, even if he was not thinking about it.
Yeah.
And on sites like Viral Zone, they do fit neatly in there.
Right. Go ahead.
I was just going to say where the listener's question is coming from is if you go to Wikipedia and you type in bacteriophage, they have a table of ICTV classification of prokaryotic viruses. And there are 17 there, but that's not classification. It's a different kind of a classification system from the Baltimore system. Yeah. So they're not in conflict, or one is not omitting the other, really.
The Baltimore is on its own, trying to classify according to how the genome is expressed. And then there's the other classical Linnaean scheme that we've often talked about with families and genera and species and so forth. That's separate. But the nice thing about Viral Zone is that it has both. It's got the Baltimore scheme, and then you can click on the type of genome, and it will give you the classical way that those are organized as well. So check that out. Phil, check it out, Phil. Kathy.
Oh, too many windows are open. There we go. All right. Robert writes, listening to your discussion in TWiV 404 about archiving and data formats recalled this mind-boggling story I saw recently. A new report from scientists Mark Zeman, Yotam Aaron, and Assam Elasta says that 20% of scientific papers on genes contain gene name conversion errors caused by Excel. In a scientific article titled Gene Name Errors Are Widespread in the Scientific Literature, the article's abstract section, the scientists explain, the spreadsheet software Microsoft Excel, when used with default settings, is known to convert gene names to dates and floating point numbers. A programmatic scan of leading genomics journals reveals that approximately 1/5 of papers with supplementary Excel gene lists contain erroneous gene name conversions. It's easy to see why Excel might have problems with certain gene names when you see the gene symbols that scientists use as examples. For example, gene symbols such as SEPT2, septin2, and MARCH1, membrane-associated ring finger CH3CH41, U3 ubiquitin protein ligase, are converted by default to 2 sept and 1 mar, which are dates in the Excel format.
Right.
Furthermore, Racaniello identifiers were described to be automatically converted to floating point numbers, i.e. from accession 2310009E13 to 2.31 to the plus 13. Since that report, we have uncovered further instances where gene symbols were converted to dates in supplementary data of recently published papers. For example, sept2 converted to 2006/09/02. This suggests that gene name errors continue to be a problem in supplementary files accompanying articles. Inadvertent gene symbol conversion is problematic because these supplementary files are an important resource in the genomics community that are frequently reused. Our aim here is to raise awareness of the problem. And there's a link to this.
To the article.
And Bob writes, always enjoy your shows, though I think the concentration on the weather is useless, froth, and makes me very impatient.
So this is really the responsibility of the investigator to make sure it's not converting automatically, right?
Right.
Yeah.
Or to not use Excel for genome data.
Yeah.
But that's a bigger problem.
I mean, it is convenient for large amounts of data, right?
Well, there are better systems for it. In fact, if you have large amounts of data, you definitely shouldn't be using Excel. You should be using some scripting language like R.
Mm-hmm.
But
R. Are you a pirate?
But people use yes, right. No, people use what they're familiar with,
of course. Aren't there other programs for big data though, besides R?
Yeah, there's Amazon has their system that's for machine learning algorithms and big data manipulation. I forget what it's called, but and you can do it in other programming languages like Python or something, but yeah, people have Excel sitting around, and so they open up their stuff in Excel, and of course it's built for summarizing the annual reports for your company, and there are some decisions that it makes that it shouldn't.
All right.
Microsoft has much to answer for.
Yeah. Yeah, unfortunately, it's pervasive, right? Everybody just uses it.
Yes.
Next one's from Anthony, who asks, was this study ever done? I only came across the original story recently, and he provides a link to an article. Actually, Kathy, you put this, or maybe he did, this BBC article.
From 2014 is what I added, yeah.
Cats and dogs to be tested for mysterious MERS infection. And Anthony says, MERS does not have the haze of the apocalypse that surrounds Zika or Ebola. Even so, anything involving the Middle East in general and the Kingdom of Saudi Arabia in particular is very, very important. Perhaps there can be continued coverage on TWiV. So we recently, Alan and I talked with Marion Koopmans and Felix Drexler quite a bit on TWiV 413 about MERS. And cats and dogs did not come up.
No.
It's mainly camels. I had a nice conversation last week with Vincent Munster at the Rocky Mountain Laboratory. Half of it was on MERS and half on Ebola. That'll go up at some point in the future. And he talked a lot about this as well. But, the camels, wherever there are camels, they get infected, basically. And you have to listen to Munster. He really puts it well. So we have continued coverage, I think.
But in Munster's, in your conversation with Munster, you didn't discuss cats and dogs, right?
Not particularly, but I asked him if there was other reservoirs, and he said, no, it's camels. It's all about camels.
Indeed.
And I've forgotten exactly what he said, but I thought it was really cool at the time. There are lots of camels all over the world. Mm-hmm. And he said if you look, you can find antibodies to MERS in all of them. So I'm not sure that's what they're worried about. And you can't get rid of it from camels because you put new camels into a herd and they get infected right away. We talked with Marion about vaccinating camels, and she said that the people who own the camels said, my camels are fine. I don't need to vaccinate them. Whereas, so that's a vaccine for an animal that helps people. Unless the animal gets sick, they're reluctant to use it. So,
or unless a family member gets sick.
Stay tuned.
Yeah. It's a bigger herd immunity that we have to think about.
Yes.
Indeed.
Rich
or a
Condit.
different herd.
Rich Condit.
Jubal writes, it looks like a kind of Muppet monster, JP in Hawaii. And it's a link. He sent us this before. It's a link to his a weather site that shows the winds in particular around Hawaii, though you can move around. I don't see how it looks like a Muppet monster.
Maybe it did when he sent it.
Maybe it did
this
when he
is real
sent
time
it.
now.
Because it's real time.
We need a screenshot.
It kind of has.
It
kind
looks
of has
a little more.
eyes.
It looks, to me, it looks a little more like Donald Trump's comb over right now.
Yeah.
P.S. Now that you got a travel budget, come out to Hawaii. October is cheap and off season. I'll take you all sailing and snorkeling. Food is delicious too. Okay.
Well, we have a budget to do TWiV, not to sail and snorkel.
Oh yeah.
We can do that on our spare time. Well, October is gone, so we're talking about next year. Would any one of the TWiV crew accept an all-expenses paid trip to Hawaii to do TWiV?
Oh, twist my arm.
Yeah.
No, you don't want to go?
Yeah, yeah.
It's a really long flight,
Yeah, I think I could manage the really long flight.
Okay.
That'd be fine. I've done that.
Give me a
before. It's
podcast or an audiobook and I'm set.
Yeah.
In business class, it goes quick.
How big is the TWiV budget?
Dickson is you're a special guy. You get business, but
that's right.
TWiV,
That's right.
TWiV is not business class. I'm sorry.
I see.
Alan, you take the next one.
Sure. Sue Ellen writes, I hope you're going to review this paper. Here's the link. Identification of small molecule inhibitors of Zika virus infection and induced neural cell death via a drug repurposing screen. And this is from Nature Medicine. Came out back in August. We didn't do this.
No,
one.
we didn't. No. One of the many Zika papers.
Yeah, a whole bunch of Zika papers came out more or less at once. And I gather this is a fairly typical strategy people use, where there's a screening panel of all approved drugs. It's a few thousand compounds, and it's sort of the first pass that everybody makes in any kind of a drug development program for a new disease, because obviously if you get a hit in there, it's already an approved drug, which is beautiful. You can then potentially cut directly to clinical trials. And it looks like they found some possibilities, and I don't know, where that leads.
They set up a high throughput test that's a little bit on the fringe but it was easy for them to assay for Zika replication. They followed up a couple of compounds to show that it was inhibitory to virus growth and cell culture, but I don't see any animal studies or anything in here.
This is the sort of quick, cheap, and dirty experiment that you bang out to before somebody else does. and then hopefully follow up and maybe take those leads somewhere.
It is a very long road from there to therapy.
Oh yes, yes. This is the easy part. Not to belittle the paper, but it's an important step to take. But yeah,
even though they're FDA approved, there are lots of issues here. One of them being, you have to give them to pregnant women.
Yes.
So you have to do a lot of studies. They looked at 6,000 compounds. This is an interesting approach. They did 2 assays. One assay, they looked for inhibition of cell death. So the virus normally induces cell death, apoptosis, programmed cell death. And they designed a high-throughput assay to look for cells that survived after treatment with these compounds. We got some of those. And then they had another assay for virus replication. I don't know what the virus inhibiting cell death by itself does not inhibit virus replication. So I'm not sure how therapeutically useful those drugs would be.
Right.
It seems to me that if you want to inhibit virus replication, right?
Yeah, my sense was that they set that up simply as a convenient high throughput method to look for candidates.
Yeah.
Right.
And then the real test is to follow up on candidates and do a genuine virus replication test. I think they had 2 compounds that they followed up with.
That's right.
Right. You start looking for your keys underneath the lamp because that's where the light is. But
wonderful. I love that.
You need to expand.
True. I love that.
It's true.
Sue Ellen continues, I'm reading it myself, but I'm not a virologist. I'm an Oracle database administrator. This looks very promising, and since you've not only have virologists but a parasitologist on your team, I would love to hear your opinion on it. Well, you just did.
Oracle database, maybe this person would know how to not get those Excel errors.
Yes, that would be putting your data into an Oracle database and using an appropriate scripting language to access it would be better than putting it into Excel, I'm sure.
Right.
Dickson, you're next.
I am. Mark writes, hello, Vincent and crew. I'm guessing that you may receive commentary about the following chronic fatigue syndrome paper, and I'm almost 100% sure that it's been across Vincent's desk already, but I'd like to draw your attention to the following paper. Metabolic features of chronic fatigue syndrome, and it gives a reference, a link, and then metabolomics. What do they have to do with virology? Well, I think it has a lot to do with virology. From the paper, CFS is an evolutionary conserved, genetically regulated, hypometabolic state similar to Dower that permits survival and persistence under certain conditions of environmental stress, but at the cost of severely curtailed function and quality of life. In environmental stress, such as a persistent ongoing infection, perhaps. I think this paper is amazing for so many reasons, but from a virology standpoint, these researchers have identified a human immune mechanism for putting on the metabolic handbrake to hinder an infectious agent. Absolutely astounding. I really look forward to hearing you talk about this paper, and when you do, I have some conversation points. Does this fit into the general category of innate or adaptive immunity? Could the different metabolic pathways affected between men and women reflect the different biology of men and women who carry a fetus and may require a more protective / less damaging set of metabolic pathways to be regulated? Could this be a genetically conserved defense mechanism which is conserved across more mammalian species? Could there be a mouse or primate model for inducing this metabolic state? Vincent, I want to also personally thank you for your assistance in the efforts of the PACE trial. We patients are very grateful to you and the others with you who have put forth the effort into bringing the trial into the spotlight. Thank you. Best regards, Mark.
Okay, you don't have to read the rest, Dickson.
I won't, except for the definition of a Dower larva.
So I did see this paper a while ago. It's quite interesting. I noticed, so it's PNAS, and it's edited by Ronald Davis, who's a member of the Academy, and then Ron's son, of course, has CFS, very serious case of CFS. So what they did is, in this study, really interesting, they took 84 subjects from all over the U.S. and Canada, and these people had CFS by certain criteria, and then they took their serum and looked for small molecules, and hence the name of the paper, metabolomics. I presume they're using
mass spec.
Mass spec.
And they identify over 400 different chemicals in blood. And then they say, are these different from people who do not have CFS? And they find, this is what they call this hypometabolic state. They find reductions in many compounds in the CFS cohort versus control people. And then one of the major ones is sphingolipids, but also phospholipids, purines, flavine, adenine dinucleotide, remember that, FAD, cholesterol, biosynthesis, bile acids, and a number of others. So they make this, some of this looks like the state called Dower, which is a response to stress where the organism kind of shuts down as a way of protecting itself, and they suggest maybe that's what's happening in these patients. They have reduced metabolic activity as a consequence of some stress. Now, the writer is suggesting that it might be a viral infection, and it could be. No one virus has ever been identified as causing CFS, but CFS patients, they often say the first onset is a viral infection, and it could be that they get multiple infections. They have a chronic infection, which in other people is harmless, like some of the herpes viruses, but in CFS patients for other reasons is not. And it could be that the body is reacting by shutting down metabolism of sorts. I don't know how we would sort that out. I mean, an animal model would be great, but I'm not aware of any. I think you would probably want to stress animals and look at their metabolomes and see if something similar is happening, right?
Well, I think an important additional experiment would be to look at samples from patients with other chronic conditions.
Yeah.
And see if this is a specific marker for CFS, which is what you'd really hope, or if this is a byproduct of some downstream process. Kind of like all sorts of problems lead to difficulty breathing, and all sorts of problems lead to flu-like symptoms.
Yeah.
There are certain endpoints you can get to, and if you're looking at an endpoint, then that may not be all that informative. But if you're looking at a specific process that's unique to CFS, then you would be on the right track. So I would hope that they would be looking at other chronic disease populations as well.
Yeah.
So the writer says that the researchers have identified a human immune mechanism for putting on the metabolic handbrake to hinder an infectious agent. I think that is going really beyond what the authors in the paper say.
That's an inference, yes.
Yeah.
Okay, that's because they don't. We're still questioning whether there's always or even sometimes a trigger of an infectious agent, though it's a possibility. And he's using the phrase human immune mechanism in I think here, a very broad context, as is some sort of defense, as if inducing a state like Dower or whatever this metabolic state is a way of combating the infectious agent. That does go beyond the paper. I think in the paper what they've done is simply they've got some clear metabolic differences between CFS patients and others.
Yeah, and it would be tremendously informative, I think, to walk this process back, whether it's specific to CFS or not, whether it's immune mediated or not. What is the step that leads to these metabolic byproducts in the bloodstream of the CFS patients? And then what's the step before that? And if you can trace that backward through the process, you'll learn a lot, I think, about not only CFS, but maybe a lot of pathogenesis.
Yeah, I mean, you have to, it seems like there ought to be some core trigger to all of this. Tracking that down would be tough, but at least you got things here that you can measure rather than a vague syndrome.
Exactly. That's a huge step.
In fact, in this paper, they do some modeling. There's a section called "Can we use this as a diagnostic for CFS?" And they narrow it to between 8 and 12. You can't measure 400 different chemicals, right? But they do some statistics and show that between 8 and 12 would give you the right answer if a person has CFS or not. So that could translate at some point into a lab test, which would really be good, right? Now, here's some N=1 anecdotal data. So our younger son, some physicians think he has CFS. Talked about this before, and and one physician here said he has all the signs of it. It seems to coincide with, he also happens to have inflammatory bowel syndrome. And whenever he has a flare-up of that, which can be triggered by diet or something, then he gets exhausted, right? So that's a stress that's caused by this intestinal problem. And the manifestation is he gets exhausted for a few days. It would be really interesting when he has one of these to do metabolomics, during that period and between when he feels fine.
Absolutely, yes. You want the controls.
But that's N equals 1. It's anecdotal. I totally understand. But there is IBD-associated CFS. That's for sure. So I think this is quite interesting. His one question, is it innate or adaptive immunity? I don't know. Like Rich said, I don't know if we can say that yet. Right? It's too early.
And we have very specific, the words innate and adaptive immunity currently conjure up a very specific series of events. Okay, if this is a defense of some sort that you could call immunity, if in fact that's even true, it may be something like totally novel. Okay, we may be talking about a new kind of dour immunity, right?
Right.
That's interesting. He says, is it conserved across mammals? Dour happens in C. elegans, right?
Right.
So it could be even more broadly conserved.
Yeah.
Who knows?
So this is May 2016. This is pretty recent. These guys are at UCSD. Vincent, get in touch with them.
Yeah.
Why not?
Yeah, sure.
Reminds me of some wisdom that Andre Varmus shared with us while we were interviewing him.
Who was that?
Harold.
Harold
Varmus. I met Harold. Come on.
Well, he is a giant in his field.
He is.
Yes.
He said the most important thing to have in any scientific experiment is an assay. So maybe this is an assay. Maybe this is
absolutely.
Yes.
Yeah.
This is very good. I totally agree. You'll see more papers coming out with this. By the way, since we're on CFS, I just want to point out that Dave Tuller this past week just published another part in his series called Trial by Error. Dave was responsible for helping to bring down the PACE study. And there's apparently another one planned along the same lines for kids. Does exercise and cognitive assistance, what is it called? Cognitive behavior therapy help kids. The other one was an adult. I mean, here the other study in adults is shown to be useless, and now they want to do another one on kids and spend millions and millions of dollars. I just don't get it. And anyway, Dave wrote this huge article. And I mean, there's so many comments already on this. It's unbelievable. It's really a trigger. Dave is doing great stuff here. And it's him that you have to thank, Mark, because I just put it on a webpage, It's no big deal. All right. Kathy, you're next.
Sam writes, Dear Commensal TWiV, my name is Sam, and I'm a fourth-year undergraduate student at the University of Toronto studying molecular genetics. I recently listened to TWiV 331 on enterovirus D68. I was wondering, since EVD68 is a picornavirus, would it be possible to make a Salk-style formalin inactivated vaccine like IPV or the HCV, hepatitis C, vaccine? To find out whether such a vaccine is in development, I Googled enterovirus D68 vaccine. I was disappointed but not surprised to see the first hit. As enterovirus D68 deaths increase, are vaccine contaminants to blame? And it gives the link. Given that my query returned only anti-vaxxer nonsense, do any of you TWiVologists know of such EVD68 vaccine candidates? Thank you for all your great Twix podcast for all my hours of counting multivulva worms, C. elegans, at my microscope. The weather here in Toronto is 26 C, 79 Fahrenheit, 299 Kelvin, mostly cloudy with 47% humidity, 1,016 millibars pressure, visibility 15 kilometers. Cordially, Sam. So I went and looked in PubMed. I can't remember exactly. I think I just put in EVD68 vaccine, and I found a paper pretty recent in PLOS One using developing cotton rat as a model for studying EVD68, and they actually do some immunization with some injected virus and show that there's some protection from challenge, but that's accompanied with some lung pathology. They also did some passive prophylactic immunization, and that was antiviral. So in other words, they transferred serum. But the main thing is that it looks like this cotton rat might be a model for studying this, and it might lead to some more future studies with respect to vaccines.
Sam says, like the HCV vaccine. As far as I'm aware, there's no HCV vaccine. Maybe he meant something else.
Oh, yeah, I didn't even pick up on that.
Certainly Salk-style IPV. You could do this, but it's not going to be done because, there are lots of D68 infections, but most of them are pretty mild. There's a handful or fraction are associated with more severe disease, but I don't think it's enough to make a vaccine. So I'm not sure it will happen, Sam.
Right.
And by the way, to Sam and anybody else who's interested in getting information on stuff like this, Google search is okay, but you can weed out a lot of the junk by going directly to PubMed, as did Kathy. And PubMed is available to anyone. It's a public database with them.
Yeah.
That's it. It'll yield the primary literature, which may be difficult to read in some cases, but that's the real stuff.
The next one is from Leona, who writes, "Hello, TWi team. Perhaps this has been mentioned before, but if not, I want to bring your attention to the evolution or devolution of the English language that has led to the dilemma caused by the lack of a gender-neutral second-person plural pronoun in English. In fact, during Shakespeare's day, thou was the second-person singular pronoun, while the plural pronoun was you." And she gives a link to this, shakespeareswords.com, thou and you. French uses the second-person plural as a formal way to address a single person, and likely this is how you came to be used in this way in English. Both German and French make a distinction between singular and plural, and, unlike English, maintain this distinction to this day. Du and ihr in German, tu and vous in French. I would prefer returning to Shakespearean English, replacing you, your, yourself, and yours with the more precise thou, thee, thy, thyself, thine, ye, you, your, yourself, and yours.
Yeah. All right.
Until then, I am in favor of y'all. Thank y'all for all your work. Wow, can you imagine? How are thee today?
They are fine.
Yeah. So people are likely to tell you to stick it up thine.
Nice. Yeah, that's
wait a
great.
minute. These are plural? Not all of these are plural, are they?
No. There's
right. No, thou, thee, thy, thyself, thine would be singular.
Yeah.
And the you is plural.
You is you, your, yourself, and yours would be plural.
Okay, so the problem is that the plural has slopped over into the singular.
And we eliminated the original singulars. Oh. Yes.
Now, can someone explain what's the difference between thou and thee?
Got me.
Silence. The rare silence on TWiV.
That's right.
No.
That's right.
All right, let's look it up. Thou and thee. Thou and thee. What's the difference between thee and thou? They are early modern English second-person singular pronouns.
So there's already two of them?
Oh, we're deep in the grammar weeds here. Wow.
Don't you listen to a grammar podcast, Kathy? Is that one?
No, it's words. It's words.
Words. Well, there is
on Wikipedia, thee redirects to thou. This gets worse.
All right, forget it. Go listen to Grammar Girl. She's got a good podcast about that. It's very popular. Rich, you're next.
Okay. Lissa writes, great image in NPR story. Quote, human viruses are like a fine chocolate truffle. It takes only one to get the full experience. And the story goes on to discuss the how do you pronounce that? Guaiaco Culex virus, where, quote, the mosquito needs to catch several of the pieces to get an infection, end quote. Mm-hmm. They go on to quote Vincent Racaniello, my favorite virologist, quote, and with viruses, anything is possible. We should always expect the unexpected, he says. Thought a little chocolate is always a good thing to think about. Lissa. And she links the NPR story, and we did cover this in TWiV 404.
That's right.
Yes.
That's right.
Nice.
I think she just wants a book. That's why she said that, Dickson.
Could very well be.
Alan, can you take the next one?
Sure. Lee writes, my friend Siddharth Krishnamurthy works in virology at Washington University, St. Louis, where he has recently discovered over 100 RNA bacteriophages. Gives a link to that work. I thought this dovetailed neatly with your last piece on the expansion of DNA viruses, and I hope you find this research worth talking about in your next episode. I don't know much about virology, but I do know from my work in healthcare that bacterial infections suck. Between systemic infection, chronic wound care, and the deleterious effects of broad-spectrum antibiotics, bacteria represent one of the worst adverse events of hospitalization. Even simple procedures run the risk of infection. Antibacterial treatment is costly, may have side effects and allergic reactions, and, as MRSA and now gonorrhea are now demonstrating, are showing limits to their effectiveness. The prospect of being able to use this fresh and expansive palette of bacteriophages to administer a highly specific, highly effective solution to infection that can be administered prophylactically or therapeutically would revolutionize hospital healthcare. Imagine a world in which the worst risk in surgery was addiction to Norco and the hardest part of the antibiotic regimen was a single injection. I'm sure there are other positive applications, but my imagination is too limited to imagine them. I can, however, imagine a situation like in the movie I Am Legend, in which an errant virus engineered to seek out and destroy pathogenic tissues is turned into the vector for a worldwide vampire zombie epidemic, leaving only Will Smith and his faithful German Shepherd to survive and question the very pressing question of continued existence. The more practical ethical dilemmas of viral therapeutics are obviously more involved and intellectually stimulating, but the child in me is desperate to get my post-apocalyptic Walking Dead scenarios on. I hope this piques your interest. Thank you for your time and consideration. And Kathy has some links. She's already.
Yeah, so this story was from March 24th of this year, and I just wanted to check and see if it was something that I had just seen that came out 23rd of November. Is that how far
back we are on email?
No, I think the link was.
No. Okay.
Yeah, no. But a paper just came out about the invertebrate RNA virusphere, where there's discovery of 1,445 new RNA viruses, some sufficiently divergent to comprise new families, filling major gaps in RNA virus phylogeny. So a paper that just came out in Nature. And I kind of tagged it as maybe something for a pick of the week, but now I've spilled it. So
just published in November, yeah.
It's this week.
Yeah. So this, I've heard Eddie Holmes talk about this. It's cool. Here, Dickson, get ready, because I remember him saying they got all kinds of animals from all over. You got arthropods, annelids. Now here's one I don't know, Sipuncula. I don't know what that is. Mollusks, nematodes, platyhelminthes, Cnidaria, echinoderms, and tunicates. They collected those. 220 invertebrate species representing.
Sipuncula are a group of marine worms.
Okay.
Apparently.
Yeah, he was very excited when he gives this talk. He says things you never even heard of before. And they got, of course, lots of viruses from it. So it's a very cool study. And lots of new ones. So we did a. I'm sorry, Rich. One more thing.
I just wanted to say. Oh, I'm sorry. Okay, go ahead.
We did a study where, remember, they did computational biologic analysis of existing sequences, and they pulled out lots of DNA viruses. Remember that?
Mm-hmm.
Right.
A couple of weeks ago, and this is kind of a companion paper to that.
Mm-hmm.
Yes.
They identified RNA bacteriophages by looking for the RNA-dependent RNA polymerase motif. Okay, and that's how they figured them out. They got a lot of complete sequences. They also they looked for some other motif as well. I don't have it right offhand right now. And the paper that he was talking about is a paper from WashU in St. Louis from David Wang's lab that identifies the 122 new RNA viruses.
So this was actual sequencing. So, in contrast to the other study we did, which was an analysis of existing data. And I think the bottom line here is there's still a lot of viruses out there that we haven't identified.
Absolutely.
And probably we will never. It's an asymptotic thing because you just keep finding them. You can't get everybody, right?
Yeah.
Nope.
blood flukes. Dickson, they even found some in blood flukes. Schistosomes.
Yeah. They're everywhere. Like Chicken Man. Remember that show, Chicken Man?
No, I've never heard of Chicken Man.
You never did?
Oh, I don't know what you're talking about. You're talking about Chicken Run?
No, Chicken Man. It was up in Boston. Nope. It was in the echo chamber. It says, Chicken Man, he's everywhere. And then it went on to talk about Chicken Man in the episode of that day.
Dickson, can you take the next email, please?
I will. Happy to do so. John writes, Dear TWiVsters, last week you mentioned expense in response to my question about whether it would be better to take both flu mist and the flu shot. However, given the lousy efficacy of the shot and the mist, then having both might actually save money if you count 1, work hours lost from flu, 2, medical expenses, 3, herd immunity, and 4, costs from old people dying of the flu. Then having both might indeed be cheaper. Sincerely, John.
I just think The Shot in the Mist would be a great title for an episode.
The Shot in the Mist. Yes.
Well, you can't get
or
both.
The Mist Shot.
The Mist Shot. I don't think you can get both. I think you'd have to do some machination to get both. You'd have to go to 2 different health providers or something.
Yeah, well, that's right.
And then he would
2
say,
different CVSs.
They would say, have you recently been immunized? What are you going to do, lie?
Yeah. Right. Just right. Show me your arm.
or you could bend the definition of recently.
That's right.
Not in the past 30 seconds.
Lying has become socially acceptable now.
Oh my gosh, it has. And here we are, scientists who never lie.
Well, I wouldn't say we never lie, but not professionally.
Trust science, don't trust scientists.
All right, let's do one more. That's Kathy's, and that'll do it.
Stephen writes, Hello to the Fellowship of the Virus. Greetings from Pittsburgh, Pennsylvania, where it is currently partly cloudy and 25 C with a dew point of 19 C. That's 67% humidity, for those who are wondering. In regard to your discussion of varicella-zoster virus vaccines in TWiV 403, I am no expert, but I can confirm that the Merck shingles vaccine, Zostavax, does contain a higher virus titer than the chickenpox vaccine, Varivax, with very few other differences. Monographs for the 2 products are linked below, and he gives those. I've also seen the effects of the varicella vaccine wear off about 7 years after administration. My younger brother was one of the patients who got the vaccine in early 1994, not long after its launch. At the time, there was no recommendation for a booster. However, 7 years later, he got a very mild case of the chickenpox the day before Thanksgiving. Thankfully, the case resolved within a few days. I, however, was the monster when it came to varicella. I returned home from a routine visit to the pediatrician at 18 months old, carrying the virus with me, and then proceeded to give it to my 5-month-old sister and my father. They will never let me live that down. I find your podcast to be light, witty, and insightful about the world of viruses. I wanted to say thank you for helping me through a difficult transition in my life. I have recently moved to Pittsburgh to start my PhD in molecular biophysics and structural biology at the University of Pittsburgh. In order to do so, I left my job at Merck's Maurice R. Hilleman Center for Vaccine Manufacturing, where I worked with both MMR and varicella-zoster vaccines, and my longtime home. Your weekly doses of knowledge, interlaced with humor, have helped make this difficult transition easier. I now look forward to your weekly doses of humor and knowledge and to what I can learn on this journey I have just started. Thank you again for all you do, Stephen. P.S. I started listening around TWiV 390, so I have a lot of catching up to do.
Mm-hmm.
Yes.
No, they don't go out of date, right? The old
That's
TWiVs,
right.
so you can still listen and learn, but just don't listen to number 1.
No. Number 1 is a lot of people's favorite, so you should
definitely
listen to
listen to number 1. It all started there.
I like the Fellowship of the Virus. This is great.
Yeah, that's good.
Some people say, I don't know how to keep finding new ways to address you, but people always do.
It's the unlimited creativity of our listeners.
It's when you crowdsource Commensal TWiVome. That's a new one, right?
Great.
There have been a few today. It's excellent.
Yeah, I like it.
So we did have an interesting discussion of the zoster shingles vaccine a while ago, as Stephen says, and we were going to get someone on. So I wrote someone at Pittsburgh, actually, who I'd met, who seemed to know a lot about these vaccines, and I said, "You want to come on and talk about it?" And he said, "No, I'm not an expert on that." But it sounded like he was when I talked to him. So there you go. All right. We're still working on that. Before we move to picks, wow, we have a lot of email left here. It is not, what is it called? Email zero? Inbox?
No, it's not inbox zero yet.
It's not inbox zero.
It wasn't patient zero either.
Nope. Register today for the ASM Grant Writing Online course. This is a webinar which will include a broad overview of the grant writing enterprise, including writing NIH and NSF grants, biosketches, and viewing grants from the reviewer's perspective. It's a 6-part series that will take place from January through March 2017. Now, this can't be right. It says the registration deadline is February 10th, but it's already started by then. Have I been saying it for months and months? Let me look at the website. Here we go. Grant writing course. Oh, boy. The schedule is from August to September of 2017. That's next year. And the registration deadline is July 15th. It's a long way in the future. All right. Anyway, if you're interested, you're going to forget about it by the time the registration deadline comes. Anyway, bit.ly/ASMGWOC17. Registration deadline is July 10th. All right, let's do some picks of the week. Alan, what do you have?
I have this was actually inspired by Rich's picks a couple of weeks ago, the Vendée Globe, which is still ongoing. But I recalled that there's an extreme sport that I also follow and kind of tangentially participate in. It's an extremely nerdy extreme sport. And this video is a little artifact from that activity. The explanation here is one of the little sub-activities, a side game in ham radio, is seeing how many countries you can contact on the radio. And for that purpose, hams define countries a little differently from the way the UN does. So if you have an island that's distant from the mainland, it may be a possession of some other country, but it counts as a separate country because there's a different challenge in trying to contact it. Now, some of these places that count as countries for this purpose don't have any people on them. So what happens is periodically, excuse me?
Nothing. I'm just laughing. I find that.
So periodically, a group of crazy people will engage in the most bizarre form of adventure travel you can possibly imagine. They will go to one of these places, these uninhabited specks of rock somewhere that counts as a country for ham radio, and they will set up a huge set of ham radio stations and contact the world. And of course, there are millions of hams trying to contact them simultaneously, and there's this big jam up on the airwaves. And it's a lot of fun for everybody, although the definition of fun may vary somewhat. So this is a video of a recent one of these. This is the VP8 SGI was the call sign to South Georgia Island. Now, lest South Georgia give you an idea of warm weather and hush puppies, this is a very different place. This is a godforsaken rock down near the Southern Ocean, and there's nothing on it but penguins.
There's a few seals walking around.
Oh, yes, there are
cute
a few
little seals. They're like these tiny things that are walking around. These guys are setting up an antenna. They're looking at them, going,
So they make this horrific journey there and land on the beach and haul their equipment up the rocks and set up these stations. And a few minutes into the video, they've got a blizzard coming through and the tent walls are blowing down.
Did you call them, Alan?
I did. I did not get through to this particular expedition. I wasn't quick enough on the draw. They were almost done by the time I got into the pile-ups. But anyway, this is very entertaining to watch. It's not something I would participate in on that end.
Table is falling over. All their computers are falling.
That's a great one. The GoPro video of the guy getting knocked over at the station.
This one guy has a Heil Pro headset like yours.
Oh, yeah. Yeah, Heil's very popular in ham radio. So
I think these people have a lot of time on their hands, huh?
They have a lot of time and a huge pile of money.
Wow.
They get some sponsorship from equipment manufacturers, but most of these guys write checks for tens of thousands of dollars for the privilege of going on this kind of a trip.
Lord.
What does 73 CU mean?
Oh, that means best wishes. I'll see you later.
Amazing what people do. Thank you.
Yes.
Rich, what do you have?
I have just a little ditty I found here on Facebook called The Dance of Earth and Venus Around the Sun. And it's an animation that it's from a site called The Bright Side. There is a website and a Facebook site for this outfit, and they just post fun stuff of all shapes and sizes, some scientific, some totally other stuff. At any rate, what this is, the Earth orbits the sun, or Venus orbits the sun 13 times for every 8 times that Earth does. And if you draw a line between them and track their orbits, and at intervals lay down the image of the line, the axis between them, you get a cool pattern. Oops. And that's all that is.
Spirograph.
It's a spirograph. Mm-hmm. That's exactly what it is. And in fact, the more I thought about it, the more I thought, well, this is a little cooked, okay? Because in fact, the orbits are elliptical, and any two things that you designed to do this would give that pattern. But what the heck? It's fun. It is sort of more or less happening out there, and I thought it was pretty.
There's one thing on this site, Rich, how you look after 1, 2, 3 glasses of wine. It's really funny because they take 4 pictures of people, men and women, and it's just so weird. They get all smiley and funny, and that's really funny. Oh my gosh, no more wine. That's it. I've had it.
There you go.
So do they still make spirographs? Oh yeah, I guess so. I just found it on the
Oh yeah. My daughter got one, and I was thrilled when she got the package, and it was just like the one that I had when I was a kid. It was the same packaging and pens and everything.
Yep.
This Earth and Venus thing has some very interesting science history behind it as well, because transits of Venus across the sun visible from Earth only occur in pairs every, I think, 108 years. And we just had a pair of them that concluded a few years ago. Previous pairs of transits were used to establish major astronomical measurements. So yeah, there's been a lot of neat science triggered by that particular pattern.
Sure.
Oh, I love this series. Find differences between these pictures. I used to love those things. Remember?
Oh yes.
Great. These are great.
Where is Waldo?
This is the good time waster, Rich Condit.
Yeah. By the way, I need to give you the Vendée Globe update. Yes. Alex Thompson, the British sailor, is still in the lead by a mere 31 nautical miles, though the third place guy is back 320 nautical miles. The leaders are now in the Southern Ocean. Excuse me?
Didn't Thompson lose a foil?
He did. He ran into a UFO, unidentified floating object. As a matter of fact, 3 sailors have been knocked out of the race. One lost the top of his mast, and 2 others hit floating objects and had to retire from the race. And Thompson lost one of his foils, this sort of outrigger that gives him some lift. So he can only get the benefit of that on one tack, not on the other tack. At any rate, they're past the Cape of Good Hope and sailing through the Southern Ocean, which is really the nasty part of the race, and he's still holding out in the lead.
All right. Dickson, what do you have?
I have the latest addition to the, oh my God, how does nature work? Biomimicry, and it's a spider that has apparently, through natural selection, of course, not through any divine intervention, ended up looking exactly like a dead leaf. How the hell does that work?
Very slowly.
I do not understand any of this process. I want to see all the intermediate spiders that didn't make it.
Well, that's the problem. You need to have the intermediate.
You do, but look at the end result. This is absolutely incredible. absolutely incredible.
Wow.
I'm just stunned.
Yep.
Wow.
Very cool.
Just one picture. That's all. That's all it takes.
Kathy, what do you have?
I picked a site. Well, first I got into it because of the Cultivar series, which is an arrangement, a beautiful set of photographs of arrangements of tomatoes, cucumbers, peppers, beans, the seeds, and beans, the pods, and various squashes and brassicas.
It's gorgeous.
And just really, it's really neat. And if you click on some of them, they have the cultivar index, and they just list all the different kinds of tomatoes or cucumbers or whatever there are. And then I realized that there was more to this site than that. And so I went to the main page of the site, and I just put in for your enjoyment the Mutatos site, which is a whole bunch of weird fruits and vegetables.
That's a riot. That is a riot.
And then overall, his thing is it has to do with what we get in supermarkets versus what there is out there in nature.
Well,
and so that's kind of his underlying theme, but just kind of an interesting gallery of things. For instance, this one called Supernatural is how different grocery stores have kind of shown utopian, supersymmetric, oversaturated landscapes. And I don't know, it's just a very interesting site also to get lost in.
I like the ultraviolet schnitzel here.
Yeah,
that shows you what a piece of meat looks like under various sorts of lighting they use in supermarkets to enhance the appearance of meat.
Yeah,
weird.
Yeah,
Lord,
interesting stuff. Yeah, I was just looking at the one about the barn. There's the iconic barn used on American advertising. So someone rebuilt a little one and drove it through several states, and now it's installed somewhere, kind of like an art thing. And they have pictures of it here. It's very interesting.
Right.
My pick is another YouTube video to follow up on last week's. Last week's I picked the
it's amazing. This is amazing.
The snake versus the what the hell was that?
It was an iguana.
Iguana. So
right,
yesterday after Thanksgiving, we had some people over, and I said, you have to watch this. So I showed it to them. And then the next one that popped up is my pick today, Kung Fu Mantis versus Jumping Spider. So this is just wonderful.
Standing.
I've never seen a mantis that's red and black. It's just gorgeous. It's got an incredible head, and it's difficult and short life.
Yes.
Yeah, I mean, it gets away from a spider, but then kind of meets its match. But again, great photography. And someone said, gosh, the people filming it must have been bored. And I'm thinking, no, I'd love to just sit there waiting for this stuff to happen.
Oh, yeah.
That would be cool. It's really lovely. This is a beautiful mantis, though. Is it called the Kung Fu Mantis, or that's just because it looks like it's doing Kung Fu? Does anyone know?
I think it's called that.
So at the end, it comes up against this huge white mantis.
Oh, don't spoil it.
I won't spoil it.
It has a duel with a jumping spider.
Yeah, it's great.
Sort of.
It's cool. Lovely video. Boy, this is great stuff that you can do nowadays. All you need is a place to go. All right, that is TWiV 417. You can find it at iTunes at microbe.tv/TWiV, and also on your iPhone or Android device. There are plenty of programs or apps that will grab podcasts for free, so just grab us and listen up. We love getting your questions and comments. Send them to twiv@microbe.tv, and consider supporting our work. You can make a contribution, either a monthly one at Patreon or a variety of other things. Go to microbe.tv/contribute. You can find out how to help us out. Why? Well, so we can go to Hawaii, live there, or more reasonably, we can go to a local lab or something and do some podcasting. Dickson Despommier, when he's not at Fort Lee, he's at thelivingriver.org, parasiteswithoutborders.com. Yep. Thank you, Dickson.
You're welcome, Vincent.
Now that
great time.
Now that you can do a good Skype from home, you should just stay there.
You think?
Yeah.
Be nice to Dickson.
Tired of seeing me come into work every day? Is that it?
No. But if you don't, want to schlep in, just stay.
It's not a schlep for me. I enjoy coming in.
All right. Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This was a lot of fun.
Rich Condit is an emeritus professor at the University of Florida in Gainesville, currently residing somewhere in Texas. Thank you, Rich.
Excellent. Thank you very much. It's always a good time.
Alan Dove is at alandove.com. You can also find him on Twitter. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. Oh, and I have to say, some new top-level domain names were just released last week, and I bought virology.blog. How cool is that? How cool is that? I got on early because all the other ones I tried were sold. Virus.blog, microbe.blog, but virology.blog. So either one will get you to the site. I thought it was cool.
We'll get you to virology blog.
Yeah, isn't that cool?
That's right.
Nice.
It's really funny. I want to thank the sponsor of this episode, CuriosityStream. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.